Does it all come out?
Yeah.
Okay. So just to caveat this talk, I'm coming from this from a microbiological point of view. I am a microbiologist rather than working with snake venom normally. But so in our field, I'm sure many of you have heard of it. Antimicrobial resistance is a serious global issue with prediction of antimicrobial resistance to be for deaths from antimicrobial resistant infection, to be into the millions, 10 million by 2050, which would be more than cancer, diabetes, cholera, for example. But unfortunately, this is based on a prediction that every single bacteria will become resistant to every single antibiotic. So it is very inflated, but it is a very serious issue. With the highest burden predicted to be in low and middle income countries, even without this prediction, we're currently seeing really high rates of resistance there. So one of the things that we can do to try and prevent this or overcome antimicrobial resistance will be to try and develop new antimicrobials. And they're desperately needed, particularly for organisms such as E. coli, which I'm sure many of you have heard of, as well as Klebsiella pneumoniae, which cause really serious infections in people. However, the biggest issue is there's not many current antibiotics coming through the pipeline to be developed. The reason being, there's not many economic incentives for a large pharmaceutical company to actually develop antibiotics. The reason being is that it will, for one antibiotic to be found, 100,000 would have to be developed, and when they do come to market, they'll be held back and not used to prevent resistance and use them in the last case possible. So that's led to very few drugs coming to market. So and no new classes of drugs of antibiotics for well over 30 years. So early stage drug discovery projects are really needed to help boost this antimicrobial pipeline. And venom has been really researched quite extensively in terms of antimicrobial properties, whether that's in scorpions, spiders, ants, and even bees. They found antimicrobial properties. But the problem with these papers is they tend to get published and then left. Nothing really happens with development. So we're looking at venom from snake, and as you can see from this table, that there's a wide variety of snake species that have antimicrobial properties, to a very large range of bacterial species, including Staphylococcus aureus, which is MRSA, where it becomes methicillin resistant, E. coli, Salmonella, and Burkholderia, Enterococcus. These are all really serious pathogens and very scary pathogens to have resistance in. So, as I said, antimicrobial properties in snake have been looked at in quite a bit of detail, but they tend to focus on the proteins that are of high abundance in the snake venom and those that are likely and have been studied before. So there's too much focus on what we already know. For example, phospholipases, A2, these are both the acidic and basic versions. We found previously and been published that they cause membrane disruption in bacteria, pore formation, and cell wall damage. L-amino acid oxidases, they also have a similar kind of activity, whether that's by oxidizing a cofactor, which results in nucleic acid protein synthesis affected, or the plasma membrane, again, membrane disruption, or through oxidative stress, which causes membrane damage. And DNA damage and causes apoptosis. It's the three-fingered toxins, which again cause membrane disruption by binding to lipopolysaccharides or lipotecoic acid, which are really fundamental to the membrane or the cell wall of bacteria. C-type lectins, as well as celloproteinases, which again disrupt the membrane. So these are the abundance of proteins in snake venom have been very widely investigated. As I said, it doesn't go much further than this is interesting and not develop into antibiotics. So we have developed an assay, which is in a project that started earlier this year, but obviously has been very heavily disrupted with COVID and coronavirus, to see if we can screen for proteins that we know and don't know to have membrane antibacterial activity. So what we do is take the whole venom and then remove any bacterial content of that, as recently published that snake venom sacs have a microbiome, so it has bacteria growing there naturally. So we want to remove any of that, so there's no effect of that into our assays, and then test against bacteria, see if there's antibacterial activity. And then when we do that, we then will try and dialyze and remove any small components from this to just focus on the proteins, because there are antimicrobial protein peptides, that cause antibacterial activity, which are interesting and something we will focus on, but in this project we're just focusing on the proteins, and then retest just to confirm that we have antibacterial activity from the proteins. And then a talk that if anyone's in the morning session, what we also do is fractionate the snake venom into its separate constituents to try and put out the protein classes to then hopefully identify later, and then retest. So what we did, we started off with approximately 14 snakes and tested it for whole venom. Again, as I said, we remove bacteria by filter sterilising and place it into PBS so that we can have a across the board and easy comparison between the snakes, different snakes venoms, and we tested against E. coli. So in this strain, it's an E. coli that was deposited by Theodore Escherich, who was one who identified and characterized E. coli and as a phenotype, as a bacteria, and this one has never been touched by antibiotics, so it's completely naive in terms of resistance and susceptibility to antibiotics. So as you can see from the arrows, we found many snake venoms that had quite high antibacterial activity. So on the left hand side is absorbance, and this is just essentially growth of bacteria, and it arrested the growth similar to the drug ciprofloxacin, which in this case is we use because it's very potent antibiotic against E. coli. So we were quite confident we could find something, or at least proteins, that would be interesting here. So then we took this on, and as I said previously, we dialyzed out all small components to test just for proteins. And on the graph on the right, you can see that we took just the snake venoms that had antibacterial activity and retested. And what we found is that the vast majority, or most of the snake venoms had equivalent activity without the small components in the snake venom and just for protein. So we were confident that there would be a protein in there. So as I said, we fractionated and pulled out all the different fractions of the snake venom, and again had them in PBS, and again we sterilised them to make sure there's no bacterial content to contaminate. But as you notice from this graph, the antibacterial activity of a single snake, so in this case N. Mossenbecker, it's not as pronounced as the whole venom. Now this could be a number of reasons. It could be an accumulative effect of different proteins in there, or it could be that some of the proteins have a synergistic activity whereby they promote or increase the activity of something else, of another protein, to have a further antibacterial activity. But we had a wide range, so there was obviously some proteins that had no activity and some that had activity on growth. So we overlaid the trace from fractionation to see if we can identify what was in there. We used this snake to begin with because this has been well characterized of the major protein constituents. So as you can see, the acidic phospholipase and the basic phospholipases were really active against E. coli in this case, whereas the three-finger toxin, cytotoxin 1 didn't, and there seemed to be a gradual activity or increase of activity around the peaks where cytotoxin 3 and 4 would be. So we wanted to confirm whether these proteins were present, and we were picking up the right things using a combination of STS page and mass spec. So initially we ran all the fractions on STS page just to identify the protein bands that were in there first before sending to mass spec. And as you can see, there's a number of fractions that didn't actually have protein that we could identify in this case, but some protein bands that we could. So we sent those ones, the ones marked with black arrows, off to be mass spec'd. But what else we wanted to do, because we wanted to focus on potential proteins that we didn't know to have antibacterial activity, we took the fractions that seemed to have some level of activity and concentrated using a 3 kilodalton molecular weight cutoff, and we were able to identify 2 extra fractions with proteins, which then, when we retested, actually increased in antibacterial activity. So there's potential that these could be something that hasn't been looked at before. And in doing so, when we got the mass spec results back, we were able to identify cytotoxin and a venom nerve growth factor protein, which were ones that we concentrated, as well as confirmed the activity of acidic and basic phospholipases and the cytotoxin. So we were able to prove that our kind of strategy was correct, and then we went ahead and carried out the same kind of process on a less characterized snake, Melanonuca, and in this case again we overlaid the trace, and we identified the proteins and identified some which from what we've seen haven't been published with antibacterial activity, or very few papers have, with the neurotoxins and cobotoxins, which is something we're going to then take forward later on. So just to conclude, we've demonstrated antibacterial activity in the whole venom of elapids, mostly towards E. coli. Fractionation and mass spec identified protein classes involved in antimicrobial activity, and we were able to concentrate some of the fractions that seem to have antibacterial activity but could potentially with low abundance, and then be able to identify them. And so we've got a simple but iterative screening process to find these proteins. And what we want to do now, once work can be picked back up after COVID, is expand to more species of snakes, and as well as other bacterial species of interest, those particularly that have resistant mechanisms, to see if we have a similar activity. Purified proteins of interest further, or reduce recombinant proteins of interest so we can test further. And also have a look at any synergistic effects of proteins of interest, as well as other marketed antibiotics, to see if it improves their activity or overcomes resistant mechanisms. Just to acknowledge both Alice and Mark from LSTM, who have been fundamental, or were fundamental, on the project when it was going at the beginning of the year, and really helped to produce a lot of this data. Thank you very much.
Thank you so much for your presentation, Alex. We'll hand you over to Rohit.
Okay. First up, do you know toxins are bactericidal, bacteriostatic?
So it seems that when the proteins are together in a whole snake venom, it is bactericidal. However, yes, when we separate out the proteins, there could be a bacteriostatic effect. But in my experience with antibiotics, there's no real bacterial static antibiotic. It only depends on the concentration. So as the concentration of the antibiotic increases, it becomes bactericidal. So it's kind of where the concentration is in how much of the protein is present in the snake venom, or the fractions, really, that is going to be the basis for that.
Any differences in microbial activity between localities of the same species, or does the venom composition variation not have an effect?
So that's something that we're keen to test, actually. So from recent reports, it seems that the composition of proteins actually changes depending on the age, the diet, the location. So that's something that could really expand this kind of assays, where we potentially be able to test snakes from totally different countries, totally different environments, that could actually overproduce or underproduce a particular protein, that we'd be able to find more of an antibacterial activity, or identify other proteins that we weren't sure or didn't know to have antibacterial activity previously.
Just muted for a second. What would the risk of administering these drugs when you try to bacteria selective?
Sorry, you broke up there slightly. I didn't hear what you said. Sorry.
Okay, I'll try again. What would the risk of administering these drugs? Will you try to design bacteria selective toxins?
So obviously there would be a risk with using them as they are, as a protein. What we would be interested in is finding the active element of the protein. So potentially finding, so we could go through this twofold, actually, is find the active element of the protein, and then hopefully use medicinal chemistry to try and mitigate for any cytotoxic, neurotoxic effects that could have, obviously, effects on humans, as well as increasing bioavailability to improve the drug. But also we can take these proteins and then we can develop resistance in E. coli or other strains of bacteria through evolution, and through a combination of experimental evolution and genomics, we can find, and have done previously, found the targets of a drug. And then when we find the targets, we then can design drugs to that new target that we found. And that could have two avenues for drug development.
That's fine. I'll pass it back to introduce the next.
Brilliant. Thank you so much for